Okay, so the time belongs to you three. So you can ask any questions you want anytime. But not all at once.
Actually, we don't have any question regarding the material, but we were like thinking if there's any possibility that we can get to know which MCQs we did wrong the last time, so that we can
Yeah, no, if you want to know about that, come to office hours. So I didn't bring the tests with me. So this is for questions about the upcoming test. The previous tests are in my office. Probably you should come to office hours sooner rather than later, because I might run out of room and shred them. I don't know if I'm allowed to yet, but so the closer to the time that you take the test, the better. But yeah, so either office hours, or if you have class during office hours, then just set up an appointment.
But we don't have any because for other classes we can see it on the blackboard itself. Is it not possible for psychology?
Like you can see which questions you got wrong on Blackboard? I have no idea. I've never heard of this. It's my first semester. There might be a simple way of doing that. Do they show the whole test? Yeah, I'm always hesitant about that because I'm sure you're a good person, but some people are not good people. And if I do that, I think there's a good chance the tests wind up online, and then I can't use those questions again. So what I do is I do it during office hours, and even then I don't show the whole test. I'll just say, okay, you got a question wrong about whatever, cognitive dissonance, and then I explain what that is.
What is the best strategy to study for this test?
So same as last time, my biggest tip is always to test yourself on the key terms. In fact, if you look at the syllabus, I have a list of 4 study tips, and the first one is test yourself on the key terms. So the way I did it when I was an undergrad and grad student for some classes is I'd have a list of key terms, well, 2 lists. One list, just with the terms, and one with the definitions if need be, and I would just audibly test myself. So I go, okay, genetic drift, that's blah, blah, blah. Next one, okay, parental investment theory, that's blah, blah, blah. I do it in a room where no one would hear me. And then I put an X beside all the ones that I didn't know, and then learn those ones. If you keep doing that and get to the point where you can say the definitions just from looking at the terms, not looking at the definition, you're probably most of the way there. As you would have seen in the last test, all or virtually all of the questions come from the key terms, so that's, I think, the most important thing to do. Yep.
On the key terms, under the brain scan category, I saw PET. What exactly is that?
Oh, interesting. That one's not in the book, I don't think. I hope it's not on the test, but it's probably a carryover from the previous textbook. I'm going to write a note to myself. I don't have paper. PET is a strategy by which you inject radioactive material into someone's blood, and then you can measure where it goes. It's not enough radioactivity to harm the person, so it's similar to fMRI in that it gives you a video. You can also use it, I think, to measure metabolism, is I think technically what it's measuring, but if it is on the test, remind me. I'll probably give everyone a bonus mark for it because it's not in the textbook and I didn't talk about it in class. Oh, yeah, if I don't notice a hand, just say something. Yeah.
Can we just describe what the resting condition?
Good, yeah, so so when the neuron's at default, it's not doing anything, not sending a signal. The positive sodium is on the outside, and there are more negatively charged ions on the inside than positively charged. So it's at around minus 70. So when it's like that, it's at resting potential.
So negative 70 voltage is resting potential.
Yeah.
And then the threshold will be negative 55.
Yeah, about negative 55. I won't test you on the numbers, but just that it's a negative charge above the resting potential is good enough. So it's around there. So then, yeah, if it goes above there, then the magic happens.
Also, can we just talk about the nodes of Ampere?
Sorry?
It's one of the terms to say nodes of Ampere.
Oh, I must have forgot to mention those. I'm quite sure they won't be on the test, but it's the gaps between the myelin sheath. So the myelin sheath are those bits of fatty tissue around the axon. They make the charge go faster. The little gaps in between the myelin sheath are called the nodes of Ranvier. So what the charge will do is jump across the myelin sheath and go to the nodes.
What is the difference between the existatory and inhibitory neurotransmitter?
Good, so when one neuron is sending a signal to another, to put it in personified terms, when the charge goes to the end of the axon, it causes chemicals to be sent across. Those are the neurotransmitters. If the neurons are saying, or if the neurotransmitters are saying to the next neuron, hey, send a signal, come on, send a signal, then they're excitatory. If they're inhibitory, they're saying, don't send a signal. Yep.
And then when the neurotransmitters say that, do they do it by increasing or decreasing the charge directly?
Yeah. It's through polarizing or depolarizing. So yeah, so moving the charge closer or further away from the threshold.
Yeah, so if we say the inhibitors, they say don't send the message or the signal, how would the GABA, who says inhibitory and it controls anxiety, if there is no signal is transferring, how would they control it?
Oh, so good. So I don't know if I said controls anxiety. Maybe the book says that. I may have mentioned it, but drugs that are called anti-anxiety drugs increase GABA transmission. So they lead to more GABA transmission, which slows brain activity down. Does that answer your question?
Yes.
So alcohol increases GABA too.
Can we briefly go through the neurotransmitters
Yeah, for sure. Yeah, we can go through all of them. I'll open it up so just to make sure I go in the same order that the book does. So the first one is acetylcholine in the order of the book. Acetylcholine has a number of different functions. The two main ones you should know are movement and memory. Dopamine is also involved in voluntary movement, but oh well. People with Alzheimer's disease have low levels of acetylcholine. Alzheimer's, of course, is a memory problem. Dopamine, as I mentioned, is also involved in voluntary movement. It's also involved in pleasure and the reward system. So there's a part of the brain called the nucleus accumbens that sends dopamine everywhere during experiences of pleasure. Some have argued that high levels of dopamine are associated with schizophrenia, which is a mental disorder. Low levels of dopamine are associated with Parkinson's disease, which is a movement problem.
Wait, what was that, the movement problem?
Parkinson's disease. So it's a disease in older people, and the treatment they're given is a drug called L-Dopa, which is a dopamine agonist. Next one is glutamate, so that's the main excitatory neurotransmitter. Some of the neurotransmitters can be both excitatory and inhibitory, depending on context, but glutamate is always excitatory. Again, an annoying bit of overlap. Like acetylcholine, it's involved in memory. The hippocampus generates glutamate, and the hippocampus is involved in creating new long-term memories. GABA is the main inhibitory neurotransmitter, so we just talked a bit about that. Norepinephrine is chemically similar to adrenaline, so it produces similar kinds of responses: heightened arousal, awareness, vigilance, etc. Serotonin has a number of different functions. Its most famous function is it has some connection to mood, although, as I said in class, probably not as simple as low serotonin causes depression. Some people say that. It's probably not that simple, but there's some connection to mood. And then the last one you have to know is endorphins, which are the body's natural painkillers. Yeah.
So the hippocampus makes glutamate.
Yeah.
And like, is it just the hippocampus, or like does it actually like do other things?
Are you saying do other parts of the brain make glutamate? I don't know. I would suspect so, but I'm not sure. I know the hippocampus generates it, and that what that does is so the hippocampus sends signals out, and it strengthens the connections between neurons in the cortex, which is how memories are stored. But as far as whether any other parts of the brain make glutamate, I would bet that they do, but I don't know for sure.
Then for the other neurotransmitters, do they also have like specific places where they're made?
Yeah, so dopamine I know is at least partly made in the substantia nigra, which is, I'm thinking back to my neuro class, I think in the midbrain, somewhere near there anyways. And I'm sure other places too. That's the only one that comes to mind. But yes, there are often specific parts of the brain that make. Oh, and then the other famous one is the pineal gland makes melatonin, which is a neurotransmitter involved in sleep. Those are the only ones that come to mind, but Wikipedia, I'm sure, knows more. Yep.
So in excitatory and inhibitory neurotransmitters, is that related to stimulants and depressants?
Sometimes. So I mean, dopamine I believe is primarily excitatory, although I don't think it's exclusively excitatory like glutamate is, and the one stimulant is cocaine, which acts on dopamine. It gets complicated because sometimes inhibitory neurotransmitters actually serve an excitatory role in the brain overall. So sometimes one part of the brain is inhibiting another inhibitory part of the brain. So the definition of stimulant versus depressant has to do with its overall effect on the brain. But off the top of my head, yeah, like cocaine acts on dopamine, which is excitatory, and that's a stimulant. Alcohol acts on GABA, and it's a depressant, and GABAtorol is an inhibitory. So there's a kind of connection there, but that's all I could say. I will say there is actually a whole course here called Drugs and Behavior. Drugs is not my area of expertise, but there is a class here. Sometimes my students are disappointed because like I've never done drugs. I have no like funny drug stories involving myself. So that class may be of interest. Yep.
What is spinal reflexes?
What is what?
Spinal reflexes as well.
Good. So yeah, reflexes, spinal reflexes are movements that are generated by the spine, not the brain. So most of our movement is generated by the brain. So the brain sends signals through the spine to the muscles. But a spinal reflex is, the example I used in class is if you touch a hot stove, typically your hand will move away before you're even aware of the pain. The spine is, quote, smart enough to detect that there's bodily damage, and it can initiate a retraction even before the brain has processed it.
So fine if you just describe through the central nervous system, like the branches of it.
Yeah, for sure. The branches of the central nervous system, you said? So, well, broadly it's branches of the nervous system. So, the nervous system divides into the central and peripheral. The central is just the brain and the spinal cord. So that's all that's included there. Do you want me to describe this one too?
Okay.
Yeah. So the peripheral includes the somatic and the autonomic. The somatic is conscious. So the peripheral is the nerves that go out from the spine. So the nerves in the rest of your body. The somatic is the conscious part. So the fact that I can, touch this and I feel it, or that the nerves get my muscles to move and do things, those are part of the somatic. So touch and voluntary movement, those are the nerves associated with the somatic. Autonomic means outside of awareness, or it's similar to a word that means that, right? Automatic means it's not consciously controlled. So this includes the sympathetic and the parasympathetic. Sympathetic is fight or flight, and parasympathetic is rest and digest. So parasympathetic is default, normal activity. Sympathetic is, okay, panic mode, all energy goes to the heart, lungs, and muscles. Sorry?
And you also the central nervous system.
So central is just brain, spine, that's it. Yeah. Yeah, the central one is the simpler one. Well, simpler to remember, of course. The brain is more complicated, but as far as remembering what's under it, yeah, it's just those two.
Will you ask detailed questions about like neurons?
Yeah, that's fair game, yep.
Like,
I don't know what you mean by detailed, but like, do you mean like the parts of the neuron?
yeah.
Yeah, parts of the neuron are fair game, yep.
What is
Yep.
the definition for mirror neurons?
Motor?
Mirror neurons.
Oh, mirror. Mirror neurons are neurons that activate both when you do something and when you watch someone else do something. So I'll do it for me. So my mirror neurons, when I choose to raise my hand like this, my left motor cortex lights up to get me to do this. But if I watch someone else do this, the same parts of my brain light up. So there are neurons that are designed to light up both when you do it and when you watch someone else do it.
What about like the hemisphere? Like if we see on the right visual, our left hemisphere is the one that's moving.
That's a good question. I'm not sure which one. I would assume if I watch you do it, it's the same. So if I watch your right, it's my left, I think, but don't quote me on that. I won't test you on that. Yep.
So what exactly does the pons do?
The pons has a few jobs. the cranial nerves run through it, so it's involved in modulating facial expressions. The cranial nerves connect with the face. It's also involved in sleep because, sleep and arousal I should say, because the reticular formation runs through it. So those are the two things that I think are good enough to know for the test. Yep.
And what is the hypothalamus do?
Hypothalamus is the 4 F's. So it's the boss of the hormone system or endocrine system.
What about the basal ganglia?
So the basal ganglia is a number of structures that are involved in, they have a similar role to the cerebellum. They help ensure that movement is well executed. So the motor cortex is what initiates motor commands, but the basal ganglia kind of help out. The basal ganglia includes a part of the brain that's important to know, which is the nucleus accumbens, which is, I mentioned earlier, sends dopamine.
Do we have to know the function for the fMRI, MT, and like the machines that we use?
Yep. So the most important distinction to make is the ones that give you a picture and the ones that give you a video. So MRI and CAT scans just give you a picture so you can see which parts of the brain are damaged, for example. fMRI and EEG give you videos, but somewhat different ones. EEG gives you brain waves, and it only detects them at the surface of the brain. It's harder to make inferences about deeper structures. fMRI gives you a 3D video. It's just a series of MRI pictures.
What is the main function of the midbrain?
The midbrain, the functions that you need to know, it's involved in simple, unconscious sensory processing and movement control. So think about blind sight. So blind sight patients have the visual cortex damaged, so the world is black for them. They have no conscious visual awareness. But if you flash a light here and ask them to guess where it is, they guess where it is more often than chance. So, you know, if there's like a loud bang over here or a bright flash and I go like this, the midbrain's involved in that kind of thing, but not detailed conscious visual processing.
What is the blind spot?
So blind sight is a condition where people are consciously blind, but they can still be guided by vision to some extent. So to put it crudely, if I damaged your occipital cortex, the world would go black. But if I put a bunch of random objects in front of you, you could still step over them. So some of the more rudimentary visual systems are still intact, and it can still guide your behavior, but you're not consciously detecting it. Yep.
So what does smell do? Because illness does everything except smell. So what does smell do?
Well, you don't have to worry about that for this test, but we'll learn about that in the chapter on sensation and perception. It goes straight to the olfactory bulb and then to hippocampus. Actually, it's funny, this page was open. I'm reviewing my notes for the next lecture. It goes then to the hippocampus and amygdala, which may be why smell is the most evocative of the senses, by which I mean it's, I'm sure all of you have had the experience of smelling something, like a certain kind of apple pie, and all of a sudden you have memories of grandma's house or whatever. And it may be because it kind of cheats and goes straight to limbic areas like the hippocampus. Yep.
And then the amygdala, what exactly does that do?
That's an emotion center, so smell is also emotionally evocative.
Can you please describe selective breathing?
Yeah, so selective breeding is something that humans do with animals. So they, for any trait you like, so let's say, you know, you're a dog breeder and you want dogs to win races. What you do is you take the fastest ones in your litter and then you have them mate and make babies. You do the same thing with the next generation of dogs. And you keep doing that after 10 generations, you're going to have fast dogs. So you're selectively breeding them. Breeding means making them reproduce. It was Darwin who first noticed this and used it as an analogy for what happens in nature. Nature, or natural selection, is basically selective breeding, but it's not done by a conscious agent. It's just that the environment selects who gets to survive and reproduce.
Interaction for violent criminal.
Yes, this is one example that I gave of what a gene-environment interaction might look like. There's different ways this can happen. So in that example, this is from a study in my other textbook. These are cases where the kids are adopted, so they have biological parents and adoptive parents. If the adoptive father has a criminal background, so only the adoptive father, so the home environment is more violent perhaps, there is little to no extra risk that the kid will wind up engaging in criminal behavior. If only the bio father has a criminal background, there is some extra risk. By extra, I mean compared to the population average. If you're in a really unlucky case where you have both the biological father and the adoptive father have a history of violence, then you have more extra. That's not grammatically correct. A lot of extra risk. So, and it's a gene-environment interaction because the effect of having the violent adoptive father is basically nothing if the kid has no genetic risk. But if the kid has the genetic risk, having the violent home does increase the risk because this is higher than this one. Does that make sense? For whatever it's worth, just as an aside, this is a rare case where they've actually identified a specific gene. You don't need to know this for the test, but this gene was sometimes called the warrior gene in the popular press. That's probably not the right way to think about it, but there's a similar effect here where if you have this gene but don't have an abusive home, your risk of violence is not very high. But if you have this gene and an abusive home, then your risk is quite high.
What is an artificial selection?
Sorry?
Artificial selection.
Basically the same as selective breeding.
What would
Yep.
be the difference between domain general adaptation and then domain specific?
Yeah, good. So this is maybe the hardest, and I will say this distinction is controversial in psychology. But what I said on Thursday that I thought might help is don't think of it as a sharp line, but as a continuum. So if an adaptation is domain specific, it means it was selected to solve a particular kind of problem, a particular kind of problem that was recurrent in our evolutionary past. So one example is the capacity for facial recognition. So we have structures in the brain that are devoted to recognizing face. Like, oh, that's my mother's face, that's my enemy's face. This is a very useful ability to have, and it's domain specific. It's not for fight, it's for one kind of thing. My example of a domain general adaptation was the capacity to be conditioned or to be punished or rewarded by the environment. So if I touch a hot stove, I pull my hand away and I go, okay, I'm not going to touch hot stoves anymore. If I say words to someone and they yell at me, I think, okay, I'm not going to say those words anymore. So it's the ability to be conditioned in that way is domain general. It applies to many different kinds of problems.
What is the difference between homology and homologous?
No difference.
It's the same.
Yeah. Like homology and homologous? Yes, yeah, they're related. I won't ask you to distinguish them on the test. Yeah, the distinction that does matter is homology versus analogy.
Holy and holy and
Yeah, good. So, for our word, it's polygyny and polyandry, but they're weird words to pronounce. But so poly means many. Gyni relates to the Greek word for women, and andros, andry, relates to the Greek word for man. So polygyny, which is more common than polyandry, especially in mammals, means a small number of males will have many female partners, and polyandry means a small number of females will have many male partners.
what about
Promiscuity is like it means something similar in ordinary speech. This is neither sex is picky, both just mate with everyone. So I talked about the bonobos, very unchoosy. They'll just mate with anyone. That's that system. The way I put it in class is, they're the hippies, the hippie species.
And what would be for humans? So those are for animals, right?
Sorry?
Those would be like for animals.
Well, so evolutionary psychologists think that humans are animals. One question the book talks about is, what are we? Like, which one are we like? And the book argues, and this is kind of the standard view, is that we are mostly monogamous, but a little bit polygynous. So if you look at anthropology and look at various cultures, you'll find that many cultures, including like more traditional cultures, have some amount of high status males who will have like many wives, for example. But most people pair bond with someone and invest in the kids together. And one way to think about polygyny versus monogamy, so monogamy is where a sexual partnership results in them staying together and helping out with the kids together. The more polygynous a species is, the bigger the males are compared to the females, because males will fight each other for mates. And in humans, we are pretty similar in size compared to other species, but males are still like 15 to 20% bigger. So that shows that, at least to some extent in our evolutionary history, there's been some degree of male-male competition.
What would be the definition for aggression?
Yeah, aggression, just violent behavior between animals or humans.
What is the definition for genetic drift?
Good. So genetic drift. I'll just give the example I gave in class. If there's a pond full of ducks, one species of duck, but the species is kind of variable. So some ducks have red feathers, some have brown feathers. And a small group of ducks just decide to move. They decide that we don't like this place anymore, and they go to a different pond. And the 2 or 3 ducks that move happen to have red color, and they go to this new pond and start having babies, and they fill up the pond. Now, all the ducks in that pond have red color, but it wasn't due to natural selection. It was just because the ducks that move there happen to have red fur. So it's a way that you can get changes in gene frequencies, but with no natural selection.
Can we discuss about heritability?
Yeah. Do you have like a more specific question, or just want me to,
The branch, like the shared environment.
like heritability versus shared and non-shared? Yeah, good. So these are very important terms to know. So I have a lot to say about heritability, but I'll start with just defining it, and you can ask follow-ups if you want. So behavior geneticists partition the variance into 3 buckets. So in other words, in the standard models, there are 3 sources of variation. So people are different. Why are they different? These are the 3 sources in the behavior genetics models: heritability, shared environment, and the non-shared environment. So heritability is genes. So each of these you'll get a number from 0 to 1, and then it'll add up to 1. So if, off the top of my head, personality is something like, let's say 0.4 heritable. So what that means is 40% of the differences between people in personality are due to genes. The shared environment is the environment shared by siblings. So the parents, the street, the school, that kind of stuff falls under the shared environment. So you don't have to know the number, but, well, I guess it is. There is the 3 laws that I talked about. So this effect is usually small. That's the big surprise from behavior genetics, is the effect of the shared environment is usually small. So for personality, I think it's less than 0.2, but we'll say it's 0.2. The third bucket, which is kind of hard to know what to do with, is the non-shared environment, which is all aspects of the environment that are not shared by siblings. So it can include niche friend groups, random events in your education or your life. It's really broad. It can include anything that's not shared by siblings. So the 3 laws of behavior genetics are that everything is heritable. Typically the effect of the shared environment is small, and typically the effect of the non-shared environment is large.
With the 3 laws, is there a 4th one? Because on the PowerPoint I found 4.
Yeah, some people add a 4th one, which I did mention earlier. I just didn't call it the 4th law. The 4th law, which isn't in the original paper, but someone added it later, is that everything is polygenic, meaning it's never one gene that determines the trait. It's always many genes. So for basically there's some weird exceptions like Huntington's disease, which is a psychological problem that has one gene, but almost nothing in psychology is like that. Yep.
What are subcortical structures?
What context is it? So literally what it means. So I'm trying to think if it includes things other than the limbic system. I don't think it does. So, oh, it is? Okay. So, well, probably then what's the term right before it? Is it used synonymous?
Cerebral cortex.
Yeah. So yeah. Then yeah. So the cerebral cortex is the outside, like pink, bumpy. Well, the whole brain's pink, but the outside layer of the brain. The subcortical structures are the structures underneath that. So subcortical just means underneath the cortex. So thalamus, hippocampus, amygdala, all of those would be subcortical structures.
Does the hippocampus do stuff other than making glutamate?
Yeah, so all you need to know for the test is it makes glutamate and it generates long-term memory. It's also involved in other things like spatial navigation. I think there's even a correlation between hippocampal growth and happiness or reduced depression. But the kind of intro psych things you have to know are glutamate and long-term memory. Most brain areas have multiple functions. Sorry, go ahead.
Is it doing stuff with memory by making glutamate?
Yeah, that's the standard model. The standard model is that because glutamate is an excitatory neurotransmitter, so like, this is kind of crude, but so here's the brain, and here's the cortex on the outside, the hippocampus is like here. So what it does by sending glutamate is it strengthens connections in the cortex, and those strengthened connections are how memories are stored. That's kind of the standard model of how it works.
So where are the memories are stored? Is it like just all over the cortex?
Yeah, all over the cortex is the idea. Like we like to think, oh, there's like a hard drive or something, and so I think the hippocampus is like that, but that's not how it works. It's stored as strengthened connections throughout the cortex. Probably the temporal cortex is arguably most important, especially for memories you can articulate with words. And makes sense, hippocampus is kind of encased in the temporal cortex, but other parts of the cortex matter too. What?
Reticular formation.
I still can't hear, sorry.
Reticular formation.
Oh, reticular formation. So reticular formation, it's a structure that begins in the pons and runs through the midbrain, but connects to other parts of the brain too. And it's often referred to as the brain's dimmer switch. So it can increase or decrease arousal. And one consequence of this is if you damage the reticular formation, you'll fall into a coma. So if you get more like alert or if you get more calm, the reticular formation's involved in that process.
Could you please just briefly describe about the four loops?
For sure, yeah. I mean that's important, of course, because it's on the written. So occipital is at the back. The other lobes have multiple functions, but the occipital basically has one, which is vision. And by vision, I'm referring to the early stages of conscious visual processing. So detecting color, shape, movement, the kind of basic things like that. The temporal lobe, which is here, probably the most important job is it has the primary auditory cortex, so hearing. But it also helps the occipital lobe out with vision. So the temporal lobes, if you like, figure out what something is. So is this a ball? Is this a face? Is this whatever? And one final thing, in the left temporal lobe, there are structures that are important for language comprehension, so understanding language, like Wernicke's area is what I'm referring to. The parietal lobe is up top. It also helps with vision a bit, so it does spatial analysis of visual information, so where is this? And it includes the sensory strip, which is for touch. So there, as we saw in the video, there's like different parts for different parts of the body. Right in front of that is the motor strip, which is part of the frontal lobe. So the sensory strip and the motor strip are like bordering each other. And when the motor strip begins, then we've moved into the frontal lobe. So the frontal lobe includes the motor strip, which is for moving your body. It sends commands to different parts of the body to move. The rest of the front is involved in the more distinctly human kinds of thinking. So complex reasoning, long-term planning, controlling your emotions, stuff like that.
Why does the cerebellum have more neurons and fewer neurons?
It's a product of evolution. Evolution's not around to answer questions, so I'm not sure. I mean, one thing I mentioned that maybe part of what's going on is, well, I guess that this isn't really answering your question. So as you mentioned, it has fewer connections, so it's more feed forward. But as far as why it's designed like that, I have no idea.
How do you describe byproduct?
Good, so you have to contrast it with adaptation. So an adaptation is a trait that is selected for because it is beneficial, like it benefits the organism. So like the eyes, the heart, these are traits that are beneficial. Or the simple polar bear case that I keep using, right? Like The white fur would be an adaptation. It was selected for because it benefits the animal. But adaptations sometimes have side effects. And the side effects aren't adaptive, they're just, it's just something that adaptation happens to produce. So this is just to show that not everything that evolution produces is adaptive. Sometimes it's just a side effect. So the example I used in class is the belly button, which is produced by evolution, but it's not adaptive. It doesn't do anything. It's just a side effect of the umbilical cord. The umbilical cord is the adaptation.
Can you describe about epigenetics?
So yeah, it's important to contrast epigenetics with mutations. So let's say there's a strip of DNA, with this code. A mutation is where one of the letters actually changes. So if this one changes into a T due to some process, that's a mutation. So the gene or the DNA code has changed. Epigenetics is where the genes don't change, but how they're expressed changes. So different stressors in the environment can lead to the genes expressing themselves in different ways. So the example I gave was people who were exposed to the Dutch famine, so hunger, their bodies store more fat as a result, and that can be passed on to their kids. Even though the genes don't change, the fact that their bodies store more fat still can get passed on. So it's very surprising. It's very counter to kind of the standard evolutionary models that were around before, which said that only mutations can lead to changes that get passed on.
So can we just go through
Yeah,
the three,
don't be sorry. It's my job.
three parts of neurons?
Say again.
The three sections of neurons.
Oh, 3 types of neurons? Yeah. So the 3 types are sensory neurons, interneurons, and motor neurons. You'll learn more as you go on, so you can break these down further in terms of structure, but this way breaks them down in terms of function, in terms of what they do. So in this way of classifying neurons, the middle ones are by far the most common. Sensory neurons are the ones that are connected with the sense organs. So rods and cones in the eye would be sensory neurons. So light hits them and they send signals to the brain. There are ones connected to the cochlea in the ear, where when there's vibrations in the air, they move and send signals to the brain. So that's what they do. I'll skip. Motor neurons send signals to the body to initiate movement. So in the motor cortex, for example, there will be motor neurons that send signals to the muscles to move. All the other neurons, which is most of them, will be in the middle, interneurons. So the brain is full of neurons that process the incoming signals in complicated ways. That's what these do.
There is a term that says refractory period. What's that mean?
This is in chapter 3, right?
Yes.
It's after like the action potential. Yeah. So, so when the action potential happens, so actually here, I'll do this. So this is 0. Here's the threshold, and here's the resting potential. So it goes above the dotted line, and then this happens. It starts kicking potassium out, so it goes back down. During this period, it can't send another signal. It's too busy kicking the potassium out and getting back to normal.
So we call that one
Yeah, this is the refractory period.
What do you call like the when it goes back at the end?
This?
Mm-hmm.
Nothing. You don't have to know this, but technically this part is called the absolute refractory period, and this is the relative refractory period. So here it's impossible to send a signal, and here it's just harder, but just refractory period is fine for intro.
Is it kind of this?
If you leave,
Yeah.
of course. Yep, you can leave anytime. Good luck. That's one of the recommendations I make. I stole it from another professor in the department who's also named David, David Prime. What I mean by that is just repeating something to yourself over and over again is less effective than trying to understand it and make connections to other things that you know. So, okay, think about the hippocampus, right? So you could just go, it does memory, it does memory. Or you could think, okay, well, it generates glutamate, that strengthens connections between neurons. Oh yeah, and that's related to Henry Molaison because he had his hippocampus taken out and he couldn't make memories anymore. It's kind of weird I'm using a memory example, but try and think deeply about how the hippocampus works and why it's involved in memory. I mean, if you have to, repetition is better than nothing, but if you can think of a way to make a connection and think deeply about it, that's better.
Like how the hippocampus work and
Yeah.
function.
Yeah. So that's the idea.
So the brain has two hemisphere, right? So which structures are double and which ones aren't?
All of the four lobes and most of the limbic system structures. So all you need to worry about is the four lobes that they're in half, but also the hippocampus, amygdala. The limbic system, I forget exactly. It might be that all of them are too. I know for sure the pineal gland is not, and the reason I remember that is, as we'll see later, Descartes thought that the soul talked to the pineal gland because it was in the middle and not split in half. So it might be actually that all of the limbic system structures have two. But for the test, all you need to know is the four lobes. Yep.
So does the right visual field goes to the left brain? Does that also work for hearing, like stuff going in the right ear goes to this?
Hearing, based on my memory of I guess it would have been, I forget what class it was. There was some class I took where that was talked about. I think it was cognitive. More of it goes to the opposite, but some to both. I mean, I forget the number, maybe 70-30 or something. So hearing is a bit weird. But don't worry about that for the test.
Earlier when you were talking about the sensory motor cortex, and you said that in the sensory part, once you go forward enough and you get into the motor part, that's the frontal lobe, and that made me wonder, like, how exactly were the lobes divided up? Like, why did they decide that the motor cortex is like part of the frontal lobe?
Good, so I mean, like a lot of decisions about definitions, it's partly arbitrary. If we rewound the clock, we may have done it slightly differently. The case you pointed to, though, is one of the more clear ones because there's kind of a, I'm going to forget the term, sulcus between the motor strip and the sensory strip, and there's a very clear difference in function. One has localized functions for, for movement and one for touch. The temporal lobe stands out as kind of a separate thing if you look at it anatomically, but the truth is this is partly a matter of choice, right? Like we could have divided it up differently.